Hello everyone and welcome to today's webinar, Drug Repurposing and COVID-19. Let me introduce myself first. This is Yeşim Tunçok. I have been working as a medical pharmacologist in 9 Eylül University. I'm also president of the Turkish Farmakoloji Society. This webinar is organized by Turkish Farmakoloji Society Klinik Farmakoloji Working Group. We are very happy to be with you in this virtual environment, although we prefer to be with you personally. Recent days with COVID-19 threatening all over the world, the topic drug repurposing of the webinar is very important for developing new effective and safe medications. We are very proud to have two distinguished speakers, Dr. Stefano Alcaro from Italy and Dr. Arzu Güneş Granberg from Sweden for discussing this popular topic. Many thanks to both of them for accepting our invitation. On behalf of the Turkish Farmakological Society, I would like to thank Sinem Ezgi Gülmez, who is the director of Clinical Pharmacology Working Group, for organizing this international meeting. Other thanks go to executive committee members of the Turkish Pharmacological Society for their efforts on organization of the webinar. I also would like to thank Aysun Şahin in the name of ZBox for technical organization of the webinar. For those of you just joining us from Turkey and other parts of the world, Europe, welcome again. I hope we will have a nice and interactive discussion during the webinar. After this short opening, I would like to give microphone to Ezgi Gülmez for giving her opening talk. Please, seat is yours, Ezgi.
Thank you very much indeed. Dear colleagues, dear students, dear attendees, it is with great enthusiasm and pleasure to welcome all of you to this webinar, Drug Repurposing and COVID-19, on behalf of the Clinical Pharmacology Working Group of the Turkish Pharmacological Society. I am Professor Sinem Ezgi Gülmez. I'm a medical and clinical pharmacologist, a medical doctor, and head of the executive board of the Clinical Pharmacology Working Group. Before starting our webinar, I would like to take the occasion to introduce you our Clinical Pharmacology Working Group, as we have attendees from different scientific specialties, as well as international attendees and students. The Clinical Pharmacology Working Group was established in 1999, so still a young working group and still growing. We have a lot of work to do. The new executive board members of the group have been appointed very recently indeed, in February 2020, and we're working with enthusiasm to take it to the next level. If I would summarize the missions of the Clinical Pharmacology Working Group to introduce you better, is actually to support the development and institutionalization of clinical pharmacology in our country in parallel with the developments and trends in the world, contributing to public health and rational use of medicines by expanding rational pharmacotherapy habits of physicians and candidate physicians in our country, and contributing to national and international clinical research by an interdisciplinary perspective. Clinical pharmacology is an interdisciplinary field that encompasses all components of the relationship between drugs and humans, and all clinical pharmacology professionals aim to support an improved quality of drug-oriented health services by providing teaching, research, and routine healthcare services that ensure more tolerable and more effective, suitable, safe, and cost-effective use of drugs. As you all know, we are globally experiencing very interesting times with this coronavirus pandemic. And during this challenging period, much has been and still have been discussed in the world about the effective and safe treatment and prophylaxis options for the new coronavirus disease. So the Clinical Pharmacology Working Group aims to bring forth the awareness and the universal principles to this subject. So we have organized this first webinar today. We have two special international speakers. It is my and our pleasure to introduce you also Stefano Alcaro from Magna Gracia University of Catanzaro, Department of Health Sciences, Italy, and Arzu Güneş Granberg from Novartis, Sweden, who is also Executive Board Member of our Clinical Pharmacology Working Group. Our speakers will cover this interesting topic, drug repurposing about COVID-19, and there will be a Q&A session after each talk. You can ask your questions by clicking on the red button on the right upper corner of the screen, and each moderator will ask the questions to the session speaker. And we also have good news. This webinar is being recorded and will be published on the website of the Turkish Pharmacological Society with the kind permission of our speakers. I would like to now leave the word to the moderator, Günay Yetik Anacak, to present you our first speaker and to moderate the first session. Enjoy the webinar. Thank you. Günay, the scene is yours.
Thank you, Ezgi. It is really great pleasure and honor for me to introduce my dear colleague Stefano Alcaro on a special day, the Pharmacist Day. And by this way, I would like to celebrate all the Pharmacist Day. I have found a chance to work with him for 5 years. He was the chair of the Cost Action Mutalik on drug discovery. He was really excellent leader on this area. So if I will introduce him, he graduated from Medicinal Chemistry and Technology department from University of Rome in 1990 from La Spianza. And then he get the PhD course in pharmacology in 1992. After 1 year postdoc in United States, he started to work as medicinal chemistry researcher at the University of Catanzaro, Magna Gracia. Then in 2002, he get the medicinal chemistry associate professor degree. And starting from 2011, he is working as full professor at the same university. He is author of more than 220 publications as papers on international journals and numerous communication to national and international conferences. His current age index is 39 and 41 for Google Scholar. He received grants as PI starting from 2000 and currently has coordination responsibilities for international cooperation and research projects. Among the active projects, there are the Cost Action CA 1513-5, which we had worked together. He was the chair of this action. And the PRIN 2017 on the topic of novel anti-cancer agents endowed with multi-targeting mechanism of action, whose national coordinator is Professor Alcaro. He serves as reviewer of relevant international journals, such as Journal of American Chemical Society, Journal of Medicinal Chemistry, European Journal of Medicinal Chemistry, Bioorganic and Medicinal Chemistry Letters, and Bioinformatics. So I'm really proud to call now Stefano to the stage, and it's your turn, Stefano.
Okay, so first of all, I would like really to thank the organizing committee and the Turkish Pharmacology Society for this great invitation. For me, it's really an honor because I see that this initiative is the first of, I hope, a long series, and I'm sure that will be successful, and I hope at least, because now we have really to think with this kind of activities as first to do in with a new area of a new era of the coronavirus, and especially I want to thank Günay Yetik for the really helpful collaboration in the co-section for this kind introduction. I'm here, as you know, to treat the starting argument of this webinar, that is a drug repurposing definition and principle, and I will follow this agenda, in which I will also try to share with you at the end some of our experience in the field of drug repurposing, especially using the tools that we have in hand in our lab, especially the computational tools. When we talk about this topic, first of all, it is maybe appropriate to make a definition about the drug repurposing. It is basically the discovery of new indications for approved or failed drugs, but it is also somehow a way to adjust, expand, to modify, or to find other applications for existing active compounds. We also often see many similar words, synonyms, that are related to drug repurposing, drug reprofiling, retasking, drug indication expansion, drug indication shift, therapeutic switching, are somehow, even are not exactly the same meaning, but are somehow related to this, to the same argument. It is interesting to look at the NIH, and in particular at the National Center for Advancing Translational Science, because in this institution they also divide somehow the stage of repurposing in two areas that are named as early stage repurposing and late stage repurposing. In my opinion, this is also quite interesting to look at, because it means that there are subtopics and there are sub areas to be considered in this topic. I go straight with this slide to discuss a little bit with you about the impact in science and in the pharma market that is related with the words, with the queries, drug and repurposing, or drug and repositioning, that, as you can see in this graph, is relevant because there are more than 3,000 documents that fulfill this query, and 68% of them are related to original articles. If we want to make a summary of this slide, we can say that drug repurposing, drug repositioning is a not topic, because it's going strongly into the impact in the literature. But also I want to say another important aspect that maybe my colleague that will follow my talk will say later. It is related to the impact into the pharma companies, because basing on the literature data that I found, 1/3 of approval drug repurposed generate about 25% of annual revenue in pharma companies. So this also, I think, is a relevant issue to be considered. Why? Because obviously the economical situation in the pharma industry started to be a little bit critical in, let's say, the first years of this century. I have reported here a couple of trends in which you see on the left a dramatic incoherence in the investments toward new compounds into the market. And also, if you look at the right histogram, we see that still there is no correlation, and especially there is an hard work for companies, but also for academic group, in order to to be effective and to translate into new therapeutic tools, so the efforts and the investment done. So drug repurposing could be a way, probably not the only way to follow. And before going on with the details, I also want to make a sort of comparison between drug repurposing and the full procedure, where for full procedure we consider the normal drug discovery process. Again, if we compare in terms of time, there is first of all, obviously, an advantage in the years that should go from 7 to 5, but also maybe, it could be a faster process. But I want also to stress with you that it is hard to go, with a drug repurposing process less than 3 years. This is quite important to discuss, because we are here also to talk about COVID, and as you know, we need urgently new therapeutic tools, and drug repurposing, it is a promising option, but it is hard to be very, very quick. Obviously, money is another issue that is related to the fact that advanced drug discovery steps are always or usually already covered by the existing bioactive compounds. And another important, from my point of view, link that should be highlighted is the connection with an issue, the polypharmacology and the multi-targeting drug discovery paradigm. It is, in my opinion, quite important. We, as already Gunay said, we treated this issue in the cost action that I will mention later, but basically I want also to give very few details related to the concept. In the polypharmacology, basically we try to mix in one compound multiple features that could be, in a word, synthesized by the pharmacophore feature. And so, as you see, for example, in this picture, the tentative is to combine multiple effects. But I want immediately to say that it is different, the drug repurposing, with respect to the polypharmacology and multi-targeting goal. Why? Because in the case of multi-targeting, we would like to get synergic effect in the modulation of the target involved, while in the case of repurposing, we are not necessarily related to the same synergy, because we can try also to convert totally the application of bioactive compounds for other applications, for other use. But again, there is the same, let's say, shared feature to be active against the multiple targets. And this could be, for us, an important goal, also because we experienced, as I mentioned before, in the mutual cost action in 4 years of activity. And if some of you is interested to look at the results and maybe to have also a quick idea about what we have done, can visit our website and look also at the FAD, that is a multimedia product that summarizes the activity done, as well as other additional information. Usually in multi-targeting, we would like to make a drug design. The drug design in drug repurposing is not necessary, because we have already designed the drug. We want, in any case, to demonstrate that the pharmacophore feature that are within the drug is similar somehow to multi-targeting agents. And this issue is not so easy to do, but we have many tools that can help us to do that. And the tools are obviously related to experimental tools, but also to in silico or computational tools that are those that we use in our lab and many other labs in academia and in companies. There are a lot of methods that we can consider. Now, just to focus on one of them that is relevant, I will spend a few words trying to explain you the docking that is in drug discovery quite important to do what? To predict the interaction among two molecules. Here you see a small molecule, number one, another molecule that is big, could be something like a protein or a DNA, a target, that by the docking are explored in terms of poses or binding conformations. This procedure is filtered by an energetic estimation. Here you see an equation that I will not comment, but just to explain that the estimation is done on the basis of single terms and contributions. Obviously, we are talking about a theoretical prediction, and even in experimental methods, but especially in theoretical methods, these results should be always confirmed because are approximate by definition. But can be very useful. Because with the docking, we can quickly establish if a small molecule and a big one can interact with each other, and maybe with a mechanism that can explain also why this compound is active. Now, the point is that while for small compounds, here you see one of my favorite compounds that I studied many years ago. It is a bisalkylating agent of the DNA, in which you see these arrows that basically are related to the torsional degrees of freedom. For a small compound, I said it is relatively simple to make prediction of the conformation. Even a compound like this can have 100 of conformation within the first 5 kilocal per mole above the global minimum. So, I mean, there are a lot of conformation. It is absolutely impossible to apply the same methods in case of targets like protein, in which we want to make what? To make a sort of prediction of the folding. How to overcome this problem? Obviously, by experimental methods. And here, what I want to stress, that is, in my opinion, one of the key points, there is the structural biology between what? Between chemistry from one side and pharmacology. Structural biology, fortunately, in the last decades, 3 decades, had a dramatic improvement because we have now experimental tools able to determine the conformation even of complex targets. And from this point of view, I believe that at least I have to mention the protein data bank from a site and the CanBL from the other. I will be very, very quick just to let you, understand which are the most important issues. Probably most of you know protein data bank, that is a database accessible of 3D determined structure that are certified. And in this database, you can find a lot of information that are related to targets, not only proteins, as probably the name should evoke, because we are also a lot of nucleic acid and other kind of macromolecules included. Here you see the exponential growth that demonstrate that the information, the structural information available is growing every day. And this is very important because basically now we have information about, let's say, a number of targets involved in COVID. Maybe in a year we should have many other information. So you understand that the process of the structural biology is dynamic by definition. Here just an example that I give you. It's an antioxidant compound, very well known because it's part of the food components in many of our products. Resveratrol, that here is depicted, within one of the possible targets in a precise and accurate way by experimental methods. This is, for example, a perfect starting point for a docking analysis, in which maybe we want to replace the resveratrol with something else, and look for a different drug. A few words about the KMBL. That is also an important tool you can find again on web. It is related especially to drug-like properties that are also very important, because we know that maybe a compound that can act very well as a peripheral drug maybe has the right properties to arrive, for example, in other districts of the organism, in the brain or in other part of our body, and the drug-like properties are obviously at the basis of this analysis. Now, just another consideration that maybe can help us to understand how the docking could be very useful for the drug repurposing. And I will give you a sort of a comparison: classical versus reverse docking. In the classical docking, what we do, basically one target that here you see here, the depicted as a protein, is virtually screened against a library of compounds. Okay, compounds can be also 100,000 or millions, because the computational power now allows to do this kind of analysis. Now, let's think to reverse the procedure, and to put, instead of the protein, the ligand, so the drug, or maybe multiple drugs against a library of targets. The results will be what? Will be the information that maybe is what we want to do if we like to repurpose a compound. Okay, I don't want to go into details, because eventually in the discussion we can go ahead, but just to communicate to you how important could be the in silico part, that obviously is just one step of the entire process. Now the point is where or when or what is the starting point? Well, we have many opportunities, because we have so many existing drugs that can be used. Most of them also are available, commercially, and so the reverse docking can be really applied to many different applications. Now I want to give you, in the last part of my talk, few examples done in our lab to demonstrate that it's possible also at academic level and with relatively few resources to make a drug repurposing. Few months ago, we experienced, but we didn't know anything about coronavirus, a drug repurposing of hydroxyquinoline. It is something funny, because you know that this is one of the drugs that is discussed, maybe is controversially discussed. Maybe we will consider this topic in the discussion. But in this work that was done considering the database of FDA approved drug, we end up with a link, hydroxyquinoline versus a target that is involved in cancer. And we also demonstrated with colleagues in the lab of in biological lab that there is an activity. And so we propose hydroxyquinoline, that now is, in charge also as anti-COVID agents, as a possible active compounds against some kind of cancers. This is one example. Another one that I also like to share with you is related to a totally different application in obesity. You know that carbonic anhydrases are based on several isoforms, and there is at least one, the 5A, that is related to the obesity. We have created in this case a mixture of a database from different sources. We joined together 3 different databases and performed basically the same analysis, or very similar to what we have seen before, ending up with a nice, repurposed drugs. We have 3 anti-cancer drugs that displayed in silico and also in enzymatic test. So we confirmed also experimentally, that are comparable to acetylzolamide, that is the golden standard for the inhibition of the carbonic anhydrase. And this is another example of drug repurposing. I go straight to my last part. I want to share with you another inspiration. We are from similar countries, Italy, Turkey, but all the other countries that are on the Mediterranean Sea share also many components that are related to the Mediterranean diet. We have, just before the crisis, we have created a review to look for possible components in the Mediterranean diet that should be considered as natural drug repurposing compounds against cancer. So I invite you, I don't want to go into detail, but I invite you to eventually look at this review, in which we basically applied the same concept that I said before, the polypharmacology approach, in order to look for some effects that maybe can explain why the Mediterranean diet is superior, or at least very good for preventing some activities, and also for as a starting point for additional therapeutic tools. My conclusions are summarized in few words. The first is related to the fact that the drug repurposing is obviously a great opportunity for us, for academia, as well for pharma companies. And it is not a new thing, because this concept is just now again in vogue because the COVID, but is always there. Also, there is a strong interaction and link with the polypharmacology and multi-target drug discovery paradigm. This is also very important to highlight. Computational methods can be useful to speed up this process. And finally, I think also natural compounds should be considered seriously as a starting point for this kind of process. So I think I conclude here, and I thank all of you for your attention. I just want to show you a slide of my research team that works with me in my department, but also as a part of a spin-off company that is active from a couple of years ago. That is one of the projects that we are going on with my team. So thank you again, and I think Gunay can follow me now.
Yeah, thank you, Stefano. It was really great talk. And it was, what a coincidence, just before the COVID, you catch the hydroxychloroquine. So you have a sense of, you have some kind of biosensor that catching.
No, but I didn't catch other more, let's say, active or interesting compounds that maybe we'll discuss later, because you know that there is a controversial issue on this molecule. So it was a coincidence. So I want to share with you, because it is really pertinent to the topic, but we never expect obsolete that could be useful also for other applications.
Before I have some question, but before asking my question, I would like to remind the participant, on the right corner of the right top corner of the screen, you will see a red button, Ask. So you can write your question through this button. While we are waiting the question to collect, I have some question for you. For the hydroxychloroquine, did you see any other targets? So you said that it's also possible. It may have a possible therapeutic effect in cancer. What kind of cancer, or what was the target?
The targets is reported in the slide is a receptor related to a pathway, also could be related to let's say the kinase cascade. Again, it is hard to say if can be used for cancer therapy, but at least could help. For example, to prevent cancer in some kind of malaria-related cancers. That so because basically in one compound you have multiple effects.
And I also wonder, what about the if you wonder the other COVID potential drugs that clinical research or some other clues showing that it's useful in COVID. Did you do after the vet science? Did you do the computational chemistry in psychopharmacology? What was the result?
Okay, for the COVID applications, I just want to mention that we are in charge for a project that has been submitted to the IMI consortium, and we are working hard to make good repurposed solutions for against the COVID using the information of spike as well as other targets involved in COVID. I cannot now give you more details about that, because we hope that first of all can be demonstrate the activity with our virologists that are now going to test our predictions, and if we are lucky, and especially we are able to do a good prediction, maybe we can have additional and also quickly new therapeutic agents for treating the COVID. So in this topic, unfortunately, I cannot give you more details, but we are working, yes. If this is the question, we are working absolutely on that.
There is now one question from Professor Gökçe Topal. She is asking, she is saying, thank you very much for your interesting talk, and then asking, what are your views on patent-related barriers for repurposing drugs?
Yeah, I'm not a clinician, so unfortunately for that, I can just give an opinion about that. Because I totally agree with the fact that patients are not always, not all the same. We are now talking a lot about precision medicine. That means that We should be able to analyze the metabolic profile of each patient, and this could make a big difference when you treat with a drug or with multiple drugs. So from this point of view, it's really difficult to reply. I always think that working on repurposed drug, it is a guarantee because you have at least already done some toxicology and pharmacokinetic test. And this could be useful to speed up the process and to ensure that maybe you are investing on promising agents. This is, from my point of view, an important goal. But again, I'm not an expert of clinical pharmacology, and this is just my opinion.
Actually, when you say pharmacokinetic test, I know you can guess some of the properties of the drug just by in silico pharmacology or computational chemistry, whatever you say, isn't it? Can we guess the pharmacokinetic profile of drug by computational way?
There are now among the set of the computational methods that I displayed before. Remember the slide in which there were a lot of approaches. I just discussed about docking, okay? But there are at least one or two of them that are strictly related to ADME profiling and ADME prediction. Because now it's possible also to estimate the metabolic path that a drug that is basically composed by functional groups can follow in the body. And so it is possible also to make a metabolic tree. Now, the metabolic tree is an average metabolic tree because it cannot take into account the patient, let's say, specificity that is not equal for all bodies. But at least we can predict, and this could be very useful because in some cases, if we know that reactive metabolites are produced, we can filter the compounds in order to avoid and to invest on, let's say, safe moieties. Okay, and this could be very useful to speed up the process because it is really bad after you develop for years with a lot of money drugs, discovering at the end that the toxicology is not correct or prevents any possible application. Somehow now it's possible also to compute this before and to predict.
It's great for drug discovery. Now there are some more questions. The first question comes from Aslı Ceylan, Professor Aslı Ceylan. She thanks for the great talk, and she says that it reminds me medical chemistry classes. She is pharmacist. I would like to ask what kind of repurposing activities may be related to resveratrol?
Yes, resveratrol. Resveratrol is really a multitasking, targeting issue. We have demonstrated also with another paper that there are a lot of possible applications, not only related to the model that I showed before. I can give you the reference. Eventually, I can try to find it, and maybe I can project. Let me try if I can do it, because it is, in my opinion, another interesting review about the resveratrol activities. You know that resveratrol has polyphenols in two rings, and the polyphenol part is for sure able to prevent the oxidative stress. But the oxidative stress is not, let's say, a specific activity, is let's say a generic activity, okay? The most interesting part, in my opinion, of this kind of evaluation is looking for specific enzymes or receptors that can bind the resveratrol. So let me see if I can show you at least the reference, because this can likely be useful for some that are interested in this topic. And we have done, by the way, an investment on this, because we have a line that is related to specifically natural targets, and in my opinion, it is. I mean, it could be useful. Just let me find it, and I show you exactly the reference. If I have a moment for that, maybe I can share again the screen. Because here in this paper we look for the SGK1 inhibition, but within the paper there is also something else that maybe can be useful for the readers for the query. So eventually I can here is the reference. I can give you exactly the reference, and we can also put in the chat. I don't know if it is visible.
It's visible.
So here there is actually a large analysis of the resveratrol activity that you can see here depicted in this slide, and in my opinion, it is quite interesting. Consider also another issue that is not secondary. The resveratrol, as you can see here, is a small molecule, and as a small molecule, obviously can accommodate very well in many clefts, and this maybe can explain good but also bad effect of this kind of compound.
Thank you. There is another question. If I can switch to other questions.
Sure.
There is two more questions. First, Professor Hakan Örer asked, what do you think of the intellectual property rights regarding drug repurposing as an obstacle to develop new indications? As an example, most patients were expired for old drugs, but combining them with targeting linkers bound covalently may give a second life.
Yes, it is a big issue. I'm not an expert of intellectual properties, but I know that because we have just also in the last month encountered the problems with intellectual properties for other compounds. And in my opinion, working on existing drugs already published or deposited, eventually expired in terms of patent could be a problem. Because you should demonstrate a second activity. I know that it's possible to do that, but sometimes you need also to refer to the original, intellectual property in order to avoid any conflict of interest or any legal issues. Maybe Arzu can help us later, because from the company point of view, this is a major issue, as much as I understand, as I expect. So I agree with the consideration of this participant. It is a problem that should be carefully considered in drug repurposing.
And there is just two questions remaining. Actually, the first one was remaining at the bottom. So she Demet Sinem Güden, she thanks for your valuable information. And she wants to learn about any antiviral previously used vaccines like BCG. Might be valuable treatment therapy in COVID-19?
Vaccine is what we hope a next future. I'm not an expert of vaccine, but as much as I know, also because we have started to discuss with some experts about this issue, I believe that we need at least another year to get it. And it is not sure that we can end up with a safe vaccine. So just to reply to this important issue, because vaccine could be the solution, right? It is something that we expect. As much as I know, there are at least maybe 10 or maybe less advanced vaccine projects running worldwide. So maybe we can end up with some solutions, but we have to go in parallel, at least with therapeutic drugs, that is, small molecules or repurposing drugs, and also someone says with the plasma therapy. That could be another, let's say, temporary solution. Okay.
Yeah.
So my opinion is that we cannot invest only in one of the three issues that I mentioned. We need now for COVID work in parallel and end up hopefully with many therapeutic tools.
Actually, what Demet was asking, another professor was asking too, Sibel Göksel. So let me read her question about vaccine also. They are actually wondering if there is a vaccine repurposing. She is saying, is there a way to use the drug repurposing approach to design vaccines as well, with the reference to previous vaccines? And my question after this, for example, there is a vaccine and you know that there is an antigen and it's producing this antibody. If you can take the structure of the antibody and dock it.
Well, in principle, this is possible. In practice, the technology is a little bit different because I showed you before the docking, taking into account a small molecule versus a big one. Okay, that is the drug discovery. Approach. In case of big compounds, in theory, it could be done the same, but in practice, it's not so easy because the complex of the system is much higher. So it is possible to look for specific tools for, let's say, antibody design. In silico, but first of all, I'm not expert of that, so I don't know exactly how easy it could be to do that. And moreover, I believe that by definition, the process should be much slower in terms of computer performance because the system is bigger. So in theory, yes. In practice, I don't know. It is for sure a topic that is part of the, let's say, biocomputing section. That is another, let's say, area. I'm a medicinal chemist, more related to chemoinformatics, bioinformatics, and computing. Biocomputing is probably the more pertinent area for this kind of design. But for sure, it is also possible. Yeah.
So we are now learning that all informatics, chemoinformatics, bioinformatics, artificial intelligence, everything is coming important in COVID. So there is one question from the head of the Turkish Farmakological Society, Professor Yeşim Tunçok, is asking. Actually, you can ask, but what is the success rate of a drug in the clinical state that was found to be effective in vitro studies? You can open your sound.
Yes. No. I would like to ask, maybe Arzu can answer this question also. What's the success rate if you found a drug in vitro is effective in one indication? What is the success rate in the clinical states? Which rate? 30%, 50%, 90%? Which one?
Arzu, please.
Yes, and I will talk about it. Anyway, so for what we call the conventional drug development, de novo, the success rate is generally less than 10%. But when it comes to drug repurposing, I don't have actually a number to give you, but the general consensus is that it is actually less risky to fail, because you do have, as Stefano summarized, you do have a certain knowledge about the toxicology, at least, if not the safety from the clinical trials as well. I should stop there, otherwise I'll give a lot of spoiler about my talk.
Okay, thank you, Arzu.
Okay, so we really learned lots of things. There is just 5 minutes before the next talk. If you are not tired, I have one more question.
Sure.
Stefano.
Yeah, but I'm happy to.
Before my question, okay, there is another question just came. Professor Şule Gök is asking, do you think that drugs used in the treatment of HIV for COVID-19 are promising?
Yes, this is also one hot topic, as much as I know, because there is, a company that is involved a lot in this topic, expert of antivirals, especially in HIV. So we can also say who is, but this is not the point, because we hope in any case that could be an effective tool against COVID. I believe that it is a perfect starting point for the repurposing, because obviously we are dealing with a similar, even it's not the same, but is at least a similar disease. And many considerations that allowed to design compounds active against Ebola or HIV could be absolutely transferred, at least tested, because are definitely closer than resveratrol or hydroxyquinoline, just to talk about what I said before. So I totally agree with this starting point, and likely we can get a first lead, a first bioactive tool against the virus.
Is there any network or something like database for with the result of the docking for the COVID or for the other things? So I know there are some commercial things, but for the COVID, maybe there is some free things.
Well, every day you find something new in COVID, especially in the last weeks. So I cannot say no. Maybe tomorrow there is another one, or in the afternoon there is another one. So I'm sure that a lot of things are running, and for sure these online predictions and database are available. We have to be careful, because while talking about protein data bank, we know exactly that are certified informations, okay, that are uploaded, because there is a peer review process that is linked, because each entry of the protein data bank is always related to a scientific paper. So it is a guarantee that what you get from the PDB is real, okay? I'm not sure that anything you find into the internet in COVID now, especially in this period, can be affordable. So I'm sure there is, but you have to check about the quality. This is my suggestion.
And just before the next presentation, the presenter of the next talk has a question to you. Arzu, please ask your question with yourself.
Are you sure it's me? Because I didn't ask you a question.
Oh, sorry. Okay.
It was me.
It was okay. Ezgi, please ask your question.
Please. Yeah, sure.
Well, thank you, Stefano, for the great talk. My question actually would cover the educational part. I'm wondering, since you have been working with many students, what could you suggest to the students who are interested to work on drug repurposing? What qualifications are you expecting for your as an expert on this area? Where they can start from? How they can move forward, or what could be needed for their training, continuous training and education? And thank you again.
Thank you for this question. It is also for me nice to say something about our clients that are the students. I have to say that it is absolutely important to have a good background. And a good background means that you should be a student in chemistry, medicinal chemistry, or pharmacy, or at least biotechnology. It depends on the topic that should be followed and expanded in the career. We are talking about vaccine. In that case, maybe a more oriented curriculum in biotechnology could be useful. But if we are talking about drug repurposing, for sure, chemistry degree and pharmacy or medicinal chemistry, that probably is only available in Italy and few other places in Europe, is in my opinion, the basis. Then there is for sure a PhD course, and the PhD course, there are many very good in my country, in Europe. For sure, this is the, let's say, the normal, high formation. I'm not sure about masters, because probably even I teach also in a master that is related to development of drugs. It is often based on a variety of basic degrees, so you cannot be too much, let's say, specific. But again, also that could be an option, or maybe can be complementary to, let's say, a PhD, if you want to be really, oriented in that sense. The master has sometimes an advantage that can link better with the companies, much better than maybe PhD courses, because we are experiencing, for example, also the so-called industrial PhD course, in which we establish, before starting the 3-year PhD course, a project with a company. And in that case, work very well, because often our students after the PhD can eventually have an option to be included in the company, and this could be probably one of the best occasion, but are not simple to establish for some bureaucratic things that I don't want to discuss now. So, but just this is what I think.
Thank you very much, Stefano, for great talk and for answering all the questions. Now I would like to give the stage to Sedat to introduce the next speaker.
Thank you, Günay. And I also would like to thank Stefano for this excellent talk, and Günay for the moderation. Actually, Stefano reminded us many things. First of all, the success rate in the clinical drug development is going down. But on the other hand, the cost is increasing a lot. And plus, when somebody, this concept is not new, we know. But on the other hand, this concept is getting important and important, because first of all, to develop a drug is expensive. And the second, the most important thing is our success rate is also going down. So therefore, these kind of approaches are very important for the pharma industry to survive. Because at the end of the day, when your new drug candidate does not work, believe me, and especially if you are working in the pharma industry, this is a disaster. So, by saying that, I really would like to, it's a great pleasure and honor for me to introduce my dear colleague, Arzu Güneş Gramberg. She's a medical doctor and she got her degree from Ankara University. And afterwards, she did her specialty in medical pharmacology at Gazi University. And then she went to Sweden for PhD in clinical pharmacology at the Uppsala University. And afterwards she worked for a Swedish regulatory agency. And then she worked AstraZeneca, and now she is working at Novartis Global Drug Development as a clinical research medical advisor. So we have a real good expert to discuss drug repurposing from all the angles. And I'm giving this stage to you, Arzu, and we are waiting for you. Thank you.
Thank you very much. So I will share my slides. So thank you for the nice introduction, and I must say I really feel at home, although I'm a bit far away from all of you guys. But it's a great possibility for me, and I hope I can tell you some meaningful information today about COVID-19 and drug repurposing. So, and I also have realized that some of my slides are similar to what Stefano has already told you, but a lot is very complementary. So I'm very happy about that. So this is an important disclosure from me. This presentation is solely based on my personal views, based on the review of the literature, and does not necessarily reflect the company I work for. So this is my agenda. I will tell you a little bit about COVID-19 and drug development in terms of repurposing, definitions, approaches, and the possibilities, challenges, the regulatory aspects I realized many of you are a bit curious about. And then I would like to provide some examples, at least, for drug repurposing in association with treatment of COVID-19. So, unfortunately, this year in March, a global pandemic has been declared by the World Health Organization, initially based on the cases in Asia, but then it spread to the entire world. And I actually have a very up-to-date list of numbers here. From actually, it's based on yesterday's data, but I think it's the most actual I could find today. So unfortunately, this outbreak has so far influenced millions of people in the world, causing pneumonia and a lot of complications, and up to 287,525 people died and confirmed related to COVID-19. So just a little bit of background. I know you are very well informed about all this, but anyway, so SARS-CoV-2 is the virus leading to respiratory distress syndrome. And it is genetically associated with the SARS-like bat virus that we have heard before, but this one is actually spreading between humans, from human to human. But originally, it is thought that it came from bats. So the coronavirus comes from, as a terminology, comes from the spikes, as you see in this picture. The virus has spikes on it, so it gives like a corona, a crown-like figure, and that's why it's called a coronavirus, and it's a single-stranded RNA virus. So previously, in 2003, we have heard about the first SARS virus, and that caused an outbreak mainly in Asia. And a decade later, in 2012, we have heard another coronavirus outbreak. This time it was in Middle East, and currently we have the novel coronavirus influencing the entire world, actually. And this one is a highly transmittable and a pathogenic virus, leading to pneumonia and acute respiratory distress syndrome, and actually respiratory failure and mortality. This because of the spreading, so worldwide restrictions are in place, as you all are experiencing currently, and a lot of efforts are out there to prevent spreading. And when it comes to disease management, we have heard a lot about vaccine development, drug development, and the management of the disease. And in association with that, we have heard a lot about drug repurposing that we haven't heard that much before coronavirus. So what is drug repurposing? Stefano has already provided a very good summary, but I will just repeat. It's also known as drug repositioning, reprofiling, retesting, or therapeutic switching. As Stefano said, there can be nuances between these terms, but they aim the same strategy, which is a drug development strategy to identify new use for approved or investigational drugs as well, because this was what I was a bit careful about. They don't necessarily have to be failed, Stefano, I guess. It's okay that they are only investigational as well for different disease or medical condition than their original planned indication. So here we have a picture showing the conventional drug discovery and comparing to drug repurposing. So we call it de novo, from a novel entity is then developed from beginning to marketing. As you can see, there are a lot of stages here, and it takes a long time, up to 17 years now, and minimum 10 years. And it starts with target discovery and screening, lead optimization, ADME studies, development, clinical phases, and finally registration, which also takes time. And then marketing also takes time. So unfortunately, the success rate is reported to be lower than 10%. We don't, unfortunately, have a number because drug repurposing has not been as let's say intensively known or used as much as the conventional way. So that's why we cannot give you an exact number, the success rate, as the conventional one. Anyway, so drug repositioning then has some advantages that I will talk about and some limitations, but it actually shortens the drug development, and depending on the development stage of the repurposed drug, there can be actually a lot of time saved, a lot of costs saved, a lot of data and effort saved, and we will talk about them. But anyway, what is common is that you do have to show efficacy and safety for the targeted population. Anyway, and you have to go through the registration, the approval process. Those are just the same. So it just depends. And you have some of you have pointed already. There are patent issues. There are data availability issues, and so on. So I will talk more about that. So the drug repurposing approaches, also Stefano has named some, luckily only some. So mainly has 3 important steps, starting with the most important, identification of the right drug for the indication of interest. And here you can see in the slide some systematic approaches that could be used. They can be classified into experimental and computational approaches. And for example, binding assays to identify relevant target drug interactions, and phenotypic screening. These are known experimental approaches. And the computational approaches include a lot of different data-driven approaches, including signature matching, molecular docking that Stefano nicely has told us about, genetic association studies, for example. This has been really in when I have performed my PhD, and we have heard a lot, and we will talk about pluses and minuses, but this is generally combined with pathway mapping. And pathway mapping is the most used approach, I think, in terms of drug repurposing and also de novo drug development. And when it's the most relevant, I think, of all, and which you will also see in my examples a little bit, the retrospective clinical data analysis, it has been used the most in terms of historical repurposing, because here the data is analyzed based on electronic healthcare records or case reports or post-marketing surveillance data. So a lot of information can be received from this. And novel data sources, and these are generally linked with biobank and genomic data, and which are also linked to electronic healthcare records for drug repurposing. So step 2 then is the preclinical models, if you don't have that data already, but you do have to show your mechanism of effect for the relevant indication. And also the clinical efficacy and safety. I keep saying safety because depending on the target indication, you have to show your risk-benefit balance always. So possibilities and challenges with drug repurposing. This is a very, as Stefano also stated, the risk of failure is considered to be lower than de novo drug development because you probably have a certain level of information concerning the toxicology profile and maybe the safety profile of your drug and the PK profile. So it all depends where the drug that is being repurposed, how much data is available. And lower costs are also in scope and expected, and reduced time to reach the market, depending also, again, the developmental stage. And here we can also start thinking about the cost of patent issues. But anyway, the cost of clinical studies and regulatory applications are valid for repurposing too. So you only save the phase 1, phase 2 in some cases. So possibility with combining with other drugs. For example, when you repurpose your drug, so you might know more about the potential other drugs, and you might choose actually to combine your drug instead of developing a monotherapy. That's also a possibility for repurposing. And this has been, repurposing has been particularly attractive for rare diseases, because for rare diseases, the mechanism of the disease generally is not well known. Not much is known about the disease. So that's why repurposing based on clinical data and has been actually common way of an off-label use also has been the common data sources for repurposing. So otherwise, use of big data, retrospective analysis of clinical data, and use of artificial intelligence is very useful. Provides a lot of possibilities for repurposing of drugs now. And especially for the drugs in market, probably the pharmaceutical supply chain is in place and functioning. So also that is a quite big plus. When it comes to challenges, so previous examples have been largely opportunistic or serendipitous. So it was mainly discovering an off-target effect or a new on-target effect by chance or by clinical data. And failures can also occur with the repurposing at later stage then. And later clinical stage development is very expensive too. So in this case, it's not necessarily because of the safety profile, because that is largely known, one can assume, but it can be the efficacy data that is not enough. So there are a lot of technical, organizational, ethical, and legal obstacles. I guess the legal obstacles, as you have pointed previously, can be very much patent related. So the exclusivity is an important issue. I will tell you a little bit more about from the regulatory point of view when it comes to market exclusivity. But the patent issues, though, when a drug is transferred, let's say, from one company to another, so it is possible that some data is not made available, and maybe the patent is not that does not have that much time left for development, and several other technical issues can be there. And maybe when it comes to ethical issues, though, the data, the patient data can lead to a lot of ethical limitations, let's say. And there can be organizational challenges because the drug is just has not been developed from the beginning. So you need to adjust your organization to pursue further the development. So there can be challenges there too. So, and limitations in predictability, because you might, and this is a new pathway maybe you are trying to develop for, so it might also introduce a lot of limitations. And the big data, we talk a lot about artificial intelligence, big data, genome-wide association studies. And there are a lot of challenges in interpretation of the data. Sometimes you have to just replan on the way, because those data sometimes do not give you straightforward answers. So you do have to adjust and find the different steps within the same pathway and drug that target instead. And adverse event profiles and causality assessments, sometimes they are not necessarily very complete, and then you don't have that information simply for that drug. So, and some drugs are very old, you don't have all the records available. And some drugs actually have generics, and then eventually you do have the cost issues unless you really fix the patent and exclusivity. So here are some previously utilized drug repurposing approaches. I think many of these are very familiar to pharmacologists, because these are the ones that we have heard a lot of. But if you look at the repurposing approaches used, mainly they are based on retrospective clinical analysis and off-label use, the clinical data, and pharmacological analysis. So, and we have one in this list with in vitro screening of compound libraries, for example. So it has been used, and you can see in this indication, the new indication list, that not all indications are necessarily rare diseases. So, so repurposing has been used. We just hear more about it now because of COVID-19. So I guess I can simply give you the, let's say, most famous ones that we have heard a lot about. I was very impressed by minoxidil's story when I was taking pharmacology lessons, actually, many years ago. And it was a drug for hypertension, and then they have realized that actually it leads to it leads to hair growth. It improves the hair growth, and then it got this indication. And sildenafil, yes, I think everybody knows about it. It was a drug against angina, and then they have realized as an adverse event that it solves certain problems, let's say. And it got indication. And thalidomide is also a very well-known story. In 50s, it was used for morning sickness, and then withdrawn from the market because of the teratogenic events. So, and later on, they have actually realized, because of off-label usage and pharmacological analysis, that it can be used in different indications, including multiple myeloma. So that's that. I don't think I will go through the entire list, but I think one more important is very interesting here: aspirin against colorectal cancer. So this is just at the stage of a recommendation. I cannot find this indication in the label right now in, for example, Sweden. But this was also an interesting repurposing based on clinical data analysis. So the regulatory aspects, briefly, let's say, the requirements of for repurposed drugs for approval are in theory very similar. You need to have your pharmaceutical, non-clinical, and clinical trial data in terms of efficacy and safety. And this can be the pathway, the regulatory pathway to be chosen can differ. It is possible to go for a complete dossier or an abridged dossier. And depending on the type of the pathway, your exclusivity is influenced. And for example, in well-established use as a regulatory pathway, then you can use actually prior clinical data. There are some regulatory pathways you may choose, but those have shorter exclusivity. And all products have to have a risk management plan, and especially the complete dossiers, they have to have a pediatric investigation plan agreed, either a plan or a waiver. So generally, since orphan indications are also actual for repurposing, I just have taken it in. So the exclusivity is then 10 years. And if you do have a pediatric investigation plan, you get additional 2 years. So, and also for well-established substance, you have 1 year exclusivity. So with the FDA, though, the exclusivity timelines are a little bit shorter. For a complete dossier, it's 5 years. For orphan indication, it's 7 years. And for new use of a previously marketed drugs is 3 years. Why these numbers are important? Because it influences the profit the companies or the drug developer, the sponsor, let's say, will get in the end. So this is very important to keep in mind or evaluate in the early stages. So drug development under the pandemic. So we have been witnessing a substantial prioritization as we have never seen or heard before. This disease is really prioritized. So almost all regulatory agencies that we could understand the language of have published guidance documents, and they are willing to guide and provide technical assistance for the drug developers. And they do actually recommend accelerated evaluation for the treatment candidates for COVID-19. So This is a very clear prioritization, and also in the healthcare facilities, not only the care of COVID-19 patients, but also clinical research is prioritized. I work in clinical research, and that's why I know a little bit about this. There is a substantial interest also from the clinics that are very, very stressed because of the cases to participate in clinical trials and, take all the burden and deliver data. It's amazing. And I would like to talk about solidarity and collaboration. This is what impressed me the most during the pandemic. All the stakeholders somehow joined forces. There is a amazing feeling of solidarity and collaboration. So when it comes to big pharma, they joined forces. So they opened their compound libraries, at least some. So, and those compounds there in their libraries for that collaboration actually have some safety and efficacy data already. So this is very impressive if you know the pharmaceutical world a little bit. And there are every day we hear more initiation of pharma academic collaborations, and in international collaboration studies, for example, we hear that WHO is also organizing or facilitating some international collaboration studies. I think it's all going into the right direction. And support for clinical research, it has never been as strong before. We heard a lot about public and private fundraising, very strong political support. And there I would like to mention some very creative politicians even trying to inspire drug developers in terms of repurposing of disinfectants and cleaning materials for oral or intravenous use. But unfortunately, there has been cases getting inspired and being harmed. So it's important to know what you are talking about. But yeah, it's a good suggestion to think outside of the box. That's what we are talking about when we are talking about repurposing. So, and then there is a willingness, amazing willingness of participants for the trials and research, and availability of the people as it has never been. So there are a lot of, it will be very easy to, I think, then convince people to participate in clinical trials, because this is not that easy normally. So, and then on the other hand, we do have a time pressure. And the spread is ongoing, and the total global mortality is quite high actually. And the healthcare facilities, they do have their limitation, but despite that limitations, they are doing a great job. I think most of us are applauding them every evening still. And last but not least, there is an immense pressure in terms of economical consequences of the restrictions. So because of that, I think repurposing has been quite actual recently. So what did I do? Just to give you a bit of overview, I simply went in where I generally check information about clinical trials. This is one of the resources I have, and I simply searched only COVID-19, and as you can see here, it's 1,409 hit when you search it in clinicaltrials.gov. They don't have all the studies, but here you do have studies. Not all of these are repurposing. These are just clinical trials that are entered in this system. And they might have a lot of other approaches than drug therapies as well. But still, this is very impressive within some months. What I have, I think I can take a long break here. I was thinking like, how can I approach this list mechanistically? Because to have a complete list of drugs being repurposed is impossible to gather. There are libraries out there, resourced by the clinicaltrials.gov, as well as other resources, including the literature. But not all studies are published, not all results are available, not all, So somehow you have to use what you have in hand. So what I try to do is try to classify the drugs that I have, the compounds that I have seen are being repurposed or studied in clinical trials. So you see here, for example, remdesivir, antivirals and antibiotics, a lot of them, and I will tell you in my next slide what kind of mechanism of effect they have. And we do see a lot of antiparasitics also used. And immunomodulators, this was also a bit fascinating. When you have an infection, then immunomodulators, immunosuppressives are studied as a treatment. The reason is that this acute respiratory distress syndrome, actually the host immunity is getting very harmful. Actually, it's a very important component of the mortality. That is why a lot of immunomodulators are now also studied in the treatment of COVID-19. Many of them are studied in severe cases because in the clinical prognosis, you know what to address at what stage. And another important list here is the category of others. Here we can see a lot of relevant drugs that we can think of mechanistically. But on the other hand, we see dietary supplements. I don't have everything here. But anyway, we see dietary supplements. We see over-the-counter drugs and traditional drugs as well. And vitamins and this and that. Of course, the vitamin C here, I guess, in the clinics used intravenously. But this has been a very interesting exercise for me to just to find out which drugs are being repurposed. I'm not going to go in one by one because we need another webinar for that. So I just simply give you a brief list that I could prepare during this time. Mechanistically, though, so I grouped them into three that I could identify. I'm sure there are more, but one group simply is inhibition of viral entry and the cytosis, viral replication, viral protein expression, virion release. So a lot about the virus. And then we have host modifiers, as I have named. I simply express as alteration of the immune response to prevent mortality, because this has been reported in at least some mortality cases in the literature that host immunity harms a lot. And supportive treatment, a lot of those listed compounds are used in the supportive treatment for acute respiratory distress syndrome and respiratory failure, and secondary infections and other complications as coagulopathies and so on. So this is what I gathered for this webinar in terms of COVID-related repurposing. So substantial work is ongoing to develop efficient therapeutic strategies for COVID-19. Lives are lost or are in danger every day. Every day, hundreds and thousands of people are dying even. And all efforts are needed to stop the spread and improve the treatment options somehow. So these are some resources that I could recommend to you to check if you are interested, not only for repurposing, but also if you are interested in which clinical trials are there. For example, WHO has its own list of clinical trials, a registry of clinical trials. Not all of those studies you can find in clinicaltrials.gov, obviously. So I would recommend to check. And otherwise, clinicaltrials.gov actually is a good platform that I generally find a lot of information about studies. You can find the inclusion, exclusion criteria, a brief summary about the drug, and even participating centers sometimes can be relevant for some of us. And yeah, and who has actually for
I think we have some little problems of a connection, maybe.
I think we lost Arzu.
Yeah, we lost Arzu,
yeah,
but
yes.
at the end of this, her talk,
Yes.
we are lucky.
Okay, let's check if she can reconnect maybe in a minute. Okay.
Okay. Actually, she disappeared in a good time, so
Yeah.
all of us has to handle the questions. So
when did I disappear?
oh, just before making your last comments regarding.
That's okay.
Okay.
Wow, that was unexpected. Okay.
Okay. Okay, Arzu, can we see you, because now we cannot see you. Arzu?
Maybe they closed her camera because of the good internet connection.
Yeah. My camera is
Okay.
Okay.
Okay. Sorry for that.
Back.
I'm sorry.
No.
Thank
Welcome
you.
back.
Thank
Welcome
you.
Back. Would you like to take your last slide again, or how shall we move, Arzu?
The last slide is not so bad, so we can do that.
Let's do it.
So are we still on?
Yes.
So that was my last slide. Let's see if we can change the view. So it says it's not time for fact. It's a time for facts, not fear. This is a time for rationality, not rumors. This is a time for solidarity, not stigma. From Tedros Adhanom Ghebreyesus, who Director General. I really like this state from him. So that's my last.
Okay. Arzu, first of all, thank you very much. Excellent presentation, and you prepared very nicely, and you also gave us two views. First of all, this concept is not new to us, as Stefano showed. And when I go back to my university years, of course, this story always starts with thalidomide. Then it goes to minoxidil, and then one of the last important example is sildenafil. And it always goes this. But we always get this information. The way that we do repositioning is different, because we got the information from clinical trials, and as an adverse effect, then this effect gets our attention, and then we try to develop our new indication. So these are either line extension or indication extension. Also in oncology, we do a lot. For example, we start with colon cancer, it's an adenocancer, then try in different adenocancers as well. But of course, this reproposing or repositioning is a little bit different scenario. Now you have antimalarial, and now that antimalarial is effective in completely in another disease. By the way, this is the most difficult disease that we are having. It's not like SARS. It's not like something. I mean, look, I mean, we are just doing these things via web, and everything has changed so much. So before I have some questions, but before that, I would like to remind there's a button on your right side of your screen, and if you want to ask any question to Arzu, please just send us those questions. And by the way, there are 10,627 articles that are published for COVID, and 80% of them published during the last 2 months. That's also something that we have to be careful while we are reviewing these COVID literature. That's also another issue. But for me, the most important thing is now, maybe last 2 or 3 years, some people were against it with these clinical trials, but now, as you said, we have strong political support and economical support as well. And do you think that the pharma industry will continue or will start spending some budget to drug repurposing, or is it just a fashion? I mean, of course, it's fashion, but what is your opinion?
Yes, I think it became actual because we didn't have any choice. We didn't have time to wait. That's why it became as it become now. Otherwise, drug repurposing, as all of us has said repeatedly, has been used before, and I think we have learned a lot. Like about everything during this period, but also about drug development, that the possibilities are there. And in terms of collaboration as well, the possibilities are there. And repurposing is actually, there are a lot of systematic approaches to be used. You don't have to go only based on clinical data and anecdotes, like in hydrochloroquine case, for example, to begin with, and so on. All in all, in the end, you have to look in the overall data. So I think it's important to assume, it's important not to assume immediately that all drugs are studied, are working. We have to see the results. So also for chloroquine, hydroxychloroquine, we have to see the results of the prospective trials. And not only assume that it is working based on anecdotes from China or some clinical examples, because we might end up controversial afterwards when we see the overall data. I'm an ex-regulator. I cannot think any other way. Sorry. So you have to have a complete program, and you need to have a good methodology. So and I think all the methodologies now. All the collaborations now are learned, at least, and I'm hoping that they will be utilized more systematically in the future, and we need this.
Yeah, I think you are very right. First of all, we really need good planned randomized studies. That's very important, because when we look at the inclusion and exclusion criteria of the current studies regarding hydroxyquinolone or coronavirus studies, they are not the best ones, let's say. Arzu, in the beginning, actually, Yeşim asked that question to Stefano, but you have some part. Would you like to answer that question now, or shall we go back to other questions?
Is it the success rate? I think I have answered promptly there. The success rate of conventional drug development from a new target to market is said less than 10%. But I don't think we do have a good analysis of repurposing results. But I guess the success rate should be higher because there will be more data available that we have already sent. Stefano, would you like to add more here? Please, welcome to join us.
Stefano, you are muted.
Could you please unmute?
Unmute, okay.
Yeah.
So I have nothing to add in the sense that, I believe that from your point of view, you have more precise information about because simply you process more candidates. So that's so obviously I take your data as a reference. Our experience is obviously related to academic projects. We also collaborate with some companies, and I also, my feeling is that the drug repurposing is well accepted for clients. Let's use this from my point of view, because sometimes they have in hand something that for many reasons cannot improve in terms of valorization. So it is obvious that this way is a preferred way, especially for COVID that is so urgent.
Yes.
But I do believe that also for other kind of disease, it's a way to follow. Well, the chemical space is so large that you always can think about other entities.
Yes.
And this is obvious. And exploring the clinical space is at the moment just a dream, because it is too big. But maybe one compromise could be the focusing on drug purposing, and then a little bit around in terms of chemical space, in order to also make, if possible, differences useful for the regulatory aspect that you said. Because if you put, the famous methyl or whatever it is into the compound, you can have another chemical entity, and maybe this is much more accepted, especially at pharma company level. So this is what I want to just to add to what you said. So
thank you. So welcome. Yeah. The patent issues, I think, are the main obstacles to know for industrial developers, let's say. But anyway, it's a collaboration, and very promising, I think.
And thank you for saying that now with the new COVID area, we have more opportunity to collaborate.
Yes,
it's nice to hear this from you, from the company, because it is, I think, one of the little, let's say, positive aspects in this drama that we are living. So, thank you.
Yeah, thank you.
Actually, Stefano, I am also working at Teva Pharmaceuticals. I mean, for pharmaceuticals now, this collaboration is getting really important, and I think, as you said, this will be one of the good thing about COVID. Also, during your talk, you talk a lot about, first of all, efficacy, and of course, efficacy for when you are developing a drug, toxic, adverse drug reaction and drug interactions are very important as well. And Gökçe Topal from Istanbul University Faculty of Pharmacy is asking, what are your thoughts on potential for adverse drug interaction in drug combination therapy for drug repurposing? By the way, two Gökçes, Gökçe Sözer and Gökçe Topal, are thanking you for this excellent presentation.
Thank you, Gökçe. So she's asking about potential adverse event reactions, not as a particular one. Yes. Well,
because disease is different, patients are different. Yeah.
Patients are different. Yeah. I think a lot is already known. Let's say toxicology is probably already there. It depends where the drug being repurposed is standing already. So let's say that we have an approved drug in market for many years, and there we have a lot of pharmacovigilance data, and we do have some old safety data as well, maybe, but depending on the new target population, you might have a different risk-benefit balance. So it's not purely adverse events only. You always have to go for the balance, because depending on your new indication, if you think about thalidomide, and let's take that example. I think that's very clear. So when you give it for morning sickness to pregnant people, I mean, the risk benefit is horrible. But if you take it and give it to people with multiple myeloma, the risk benefit is completely different. There you can take more risks, and you can take actually risk mitigation measures. Then you can actually inform that target population that they should have contraception, strict contraception use. I mean, so I guess I can answer the question with that example. So it all depends the balance of risk benefit.
I think that's very important. Always for a good drug, everybody wants more efficacious, less toxic, but which is not happening in normal daily life. Plus, adverse events for some patient population, it's you cannot accept this adverse event, but for some patient population, it would be completely different. Okay, now we have 5 minutes left, and I really would like to thank again Arzu. It was an excellent presentation. Thank you very much.
Thanks for the possibility.
Again, thank you very much. And now I would like to give stage to Ezgi to make the closing speech. Ezgi, stage is yours. Thank you.
Thank you, Sedat. Thank you, all of you. And again, dear colleagues, dear students, dear attendees, it was indeed such a fruitful webinar on this hot topic, drug repurposing and COVID-19. I hope you enjoyed our webinar, because I enjoyed myself a lot. Drug repurposing is already a very challenging area, and especially during this period of coronavirus pandemic, it has gained more attention and importance. And I do believe sincerely that our speakers covered the content from definitions, principles to the clinical perspective, even within their limited time. So I would like to express our heartfelt thanks again to Stefano and Arzu for their presentations, for answering the questions, and for sharing their perspectives. Even myself, I have learned a lot. And thank you also to all the attendees from Turkey, from other countries, for their interest, for their time, for their active participations with the questions. And we had more than 100 attendees, live participants, even though we had almost 300 registered participants. But we do have good news. Again, I would like to remind that this webinar is recorded and it will be published on the website of the Turkish Pharmacological Society. So good news. And we just have a little request. We'll be sending a short feedback survey link after the webinar. Please do fill it out. It will take only 5 minutes for you and will support us for our future events. We hope that this first webinar organized by the Clinical Pharmacology Working Group will lead to new national and international collaborations. Thank you again to all of you. Follow us for our next events. Thank you and goodbye.
Goodbye.
Bye.
Goodbye.
Bye-bye.
Ci vediamo.
Bye.
Oh.